So today, today our topic is skeletal tissues. We'll talk about cartilaginous tissue and bony tissue. And here we can see classification of all tissues. Here we can see that tissues are divided into borderline tissues, including epithelial tissue, which provides border between inner and outer environment or between two inner environments. And also we have inner tissues. They are divided into specialized, including muscle and nerves, and all other tissues are tissues of inner environment. In the widest meaning, they are called connective tissues. They have a lot of similarities. They have cells and intercellular matrix which prevails. So a matrix prevails in the tissues of inner environment. And matrix has different properties which correspond to the features of the functions of those tissues. We have liquid or fluid tissues, blood and lymph. Their matrix, intercellular matrix, it's plasma, it's fluid. Also, we have connective tissues, connective tissue proper. Matrix of connective tissue proper, it's jelly-like fluid. And also we have skeletal tissues, bony and cartilaginous, and their matrix it's dense, and in bony tissue it's even mineralized. So those tissues, they have a lot of similarities, but properties of the matrix provide functions of those tissues, and also properties of the matrix, they correspond to the functions of those tissues. So today we'll talk about skeletal tissues, bony and cartilaginous. And here we can see a diagram which shows us structure of connective tissue, and in the widest meaning we describe bony and cartilaginous tissues as connective tissues. So let's revise main components of the connective tissue. All connective tissues have cells and matrix prevails in all types of connective tissues. Matrix has fibers and ground substance. Fibers are different, a little bit different in different kinds of tissues, and ground substance also has different properties. You remember that connective tissue proper may have collagen, elastic, and reticular fibers. In cartilaginous tissue, there are collagen and elastic fibers, and bony tissue has only collagen fibers. And ground substance also has different properties. So let's discuss all features of those tissues. So we have here fibrous connective tissue, cartilaginous, and bony tissue, and let's compare them. And let's look at their features. So we have cells. Cells have some features, some peculiarities, but cells are present in all those tissues, and they provide production of the matrix. They produce matrix which surrounds them. In fibrous connective tissue, matrix has jelly-like structure. It's jelly-like fluid. In cartilaginous tissue, it's denser than in fibrous connective tissue. So cartilaginous tissue is alike fibrous connective tissue, but matrix, especially ground substance, it's denser than in fibrous connective tissue. And in bony tissue, this matrix, it has organic part, but it's mineralized. Different minerals in organic substances, they provide the biggest density of the bony tissue. So here we can see increasing density of the matrix of connective tissues. In fibrous connective tissue, it's jelly-like. In cartilaginous, it's dense, but not mineralized. And in bony tissue, it's mineralized and very dense. And also there are fibers. Mainly collagen fibers, they are present in all three types of tissues, in fibrous, in cartilaginous, in bony tissue. Elastic fibers are present in fibrous and cartilaginous tissue, and bony tissue it has only collagen fibers. So the main components of connective tissues, they are present in all three types of the connective tissues. And you know that fibrous connective tissue, cartilaginous, and bony tissue, they provide a basis of all our organs. They provide maintenance of our body parts, and they are present, especially fibrous connective tissue, it's present almost everywhere, And cartilaginous tissue, bony tissue, they provide formation of the skeleton of our body. They provide formation of dense skeleton, real skeleton, and fibrous connective tissue. It provides formation of the skeleton of the inner organs. It maintains their part and in other tissues. So those tissues, their function, common function, it's maintenance and of the other cells and structures, and formation of the body shape and shape of the inner organs. And here we can see fibrous connective tissue. There are cells, there are fibers, elastic, collagen fibers, and ground substance between them. It's cartilaginous tissue. There are also cells of the cartilage, fibers and ground substance, and bony tissue, also cells and matrix. Here we cannot distinguish fibers and ground substance, but they are present here. They are mineralized and they provide formation of the bone. And also they have those tissues, fibrous connective tissue, cartilaginous and bony tissue. They have a lot of similarities in their cells. And difference of the cells of those tissues. Difference, they include 3 stages of cell maturation: stem cells, young cells, and mature cells. You remember that some tissues, they have difference. Difference is set of cells with common origin and common functions, and cells of the difference, they are following stages of maturation, beginning from the youngest stem cells till the old and dying cells. So difference of the connective tissues, they include stem cells, young cells, and mature cells. Stem cells, they become to be young cells, and young cells, they become to be mature cells. And stem cells, they provide regeneration and growth. Young cells also, they provide growth, and also they provide formation of the matrix. They are the most active cells of those tissues. They produce fibers, they produce ground substance. And after production of the ground substance, they become to be mature resting cells. They don't provide formation of the ground substance and fibers. They are resting cells. They provide maintenance of the homeostasis of those tissues. And let's look at those cells in fibrous connective tissue, cartilaginous, and bony tissue. In fibrous connective tissue, there are fibroprogenitor cells. In cartilaginous tissue, there are chondroprogenitor cells, and in bony tissue, there are osteoprogenitor cells. They are stem cells of those tissues. They provide regeneration and growth of those tissues. They produce matrix of those tissues. Fibrous connective tissue, it has fibroblasts. Cartilaginous tissue, it has chondroblasts. And bony tissue, it has osteoblasts. So all those cells, they produce matrix: fibroblasts in fibrous connective tissue, chondroblasts in cartilaginous tissue, and osteoblasts in bony tissue. And mature cells, in fibrous connective tissue we have fibrocytes, in cartilaginous tissue we have chondrocytes, and in bony tissue we have osteocytes. So here we can see blast and cyte. Blast, it's immature young cell which produce matrix, and cyte, it's mature cell which is resting cell, and it provides maintenance of the homeostasis. So there are main cells of the connective tissues, including fibrous connective tissue, cartilaginous, and bony tissue. And now we are going to the cartilage and cartilaginous tissue, and let's look at the features of the structure of the cartilage. So here we can see fibrous connective tissue, which has typical structure. It has cells which provide formation of the matrix, and different components of the matrix, including fibers and ground substance. It's jelly-like substance which fills the spaces between fibers and cells. And mainly ground substance has features which provide functions of the tissues. So here we can see fibrous connective tissue, and here we can see cartilaginous tissue, and this tissue has a lot of similarities with fibrous connective tissue. It also has cells. It has matrix which includes fibers and ground substance. And ground substance, it has a lot of similarities with fibrous connective tissue, but now we will look at the differences, mainly at the differences of cartilage and fibrous connective tissue. So, cartilage cells, they derive from mesenchymal stem cells. Here we can see undifferentiated mesenchymal cells, and they give rise to different cells of the cartilage. And also they give rise to fibroblasts. They are given rise to blood cells, endothelial cells, and others. And I think you know that there are two different branches of the mesenchymal cells differentiation. They may give rise to blood stem cells and stem cells of mechanocytes. Blood stem cells, they give rise to different formed elements of blood, and also some cells of connective tissue which develop from leukocytes of blood. And another branch, it's stem cells of mechanocytes. This branch gives rise to fibroblasts of the connective tissue. It gives rise to adipocytes. It gives rise to reticular cells of reticular tissue, and also chondroblasts, chondrogenic cells of the cartilage, and also osteogenic cells of the bones. They derive from stem cells of mechanocytes. So all cells, main cells of the tissues which provide mechanical properties, they derive from stem cells of mechanocytes. And here we can see formation of the cartilage. Here we can see mesenchymal cells. They have star-like shape, they have long projections, they communicate with each other, and after some mitotic divisions, they form chondrogenic cells. They become to be round in shape, and then they provide formation of the chondroblasts and chondrocytes. Here we can see cells surrounded with matrix, and then they give rise to chondrocytes, mature cells of the cartilage. Here we can see stages of the maturation of the cartilage, beginning from the mesenchymal cells, then chondrogenic cells or chondroprogenitor cells, chondroblasts, and mature chondrocytes, which form isogenic groups. And later we'll discuss how they are developed. So here we can see embryo on the early stage of this development, and here we can see somites. Here we can see somites. They are periaxial mesoderm. The most peripheral part of the somite, the uppermost part, it develops into the dermatomyotome. Dermatome, it gives rise to the connective tissue of skin, and myotome, deeper part, it gives rise to the skeletal muscles. And this part of the somite, it's called sclerotome. Here we can see sclerotomes of somites. And some cells of sclerotome, they are mesenchymal cells. Here we can see that it's less dense than dermatomyotome. So dermatomyotome is denser part of the somite, and this part of the somite, medial part and deeper part, it forms mesenchymal cells which are developing into stem cells of mechanocytes. Here we can see them, and they will give rise to the bony cells and cartilaginous cells. So they will give rise to the bones and cartilage, these cells, which are present near the neural tube and near notochord. So here I can see mesenchymal cells which will give rise to the stem cells of mechanocytes. Mesenchymal cells, they have projections, they have star-like shape, and here you can see mesenchyme. And some mesenchymal cells, they become to be chondroprogenitor cells. They become to be round in shape. Here you can see they lost their projections, and they become to be round in shape. Here you can see formation of chondroprogenitor cells. So a group of chondroprogenitor cells has been developed, and this group will form chondrogenic eyelid. So here we can see chondroprogenitor cells, and they begin to produce matrix of the cartilage. They produce this matrix and they surround themselves. Here you can see that cells, they are surrounded with this matrix, and then this matrix, it becomes to be denser, and it forms basis of chondrogenic eyelid. It's much denser than matrix of the mesenchyme. Dense particle chondrogenic eyelid is developed. They have produced matrix which fills the spaces between the cells. It's called interterritorial matrix, and then they surround themselves, and it's called territorial matrix. This territorial matrix is present near the chondrogenic cells. It's present in the territory of the cells. So interterritorial matrix between the cells, this one, and territorial matrix, it's produced later, and it surrounds cells. So it's formation of the matrix of chondrogenic eyelid and formation of the matrix of the cartilage. And here we can see result of the formation of chondrogenic eyelid. Here we can see cells, chondrogenic cells inside its matrix, and some cells they cover this chondrogenic eyelid, and mesenchymal cells they surround this eyelid. So here you can see chondrogenic eyelid, and it will form cartilage. Peripheral cells which aren't surrounded with matrix, they are chondroblasts. They are producing this matrix, but they don't surround themselves yet. And those cells which are surrounded with matrix, they are chondrocytes. They are located deeper. They are located in lacuna of the cartilage. So chondroblasts, unmature young cells, they produce matrix. They are located on the surface of the cartilage, and chondrocytes, they aren't producing matrix. They are located deeper in the cartilage. And let's look at the cartilage growth of the mature cartilage. Let's look at chondroblast. So growth of the cartilage begins from chondroblasts. Here we can see chondroblast, and let's look at this cell. What will happen to this cell? So here I can see cell of the cartilage. It's chondroblast, and it's located on the surface of the cartilage. And this cell produces matrix. It fills the space near itself, and also it surrounds itself with matrix. And when it has surrounded itself with matrix, it becomes to be chondrocyte. So it produces interterritorial matrix, and then it surrounds itself with territorial matrix. And now the cell becomes to be chondrocyte after production of the matrix which forms the walls of lacuna. So here we have lacuna, cavity in the cartilage, and cell inside this lacuna. It's territorial matrix which surrounds the cell, and it has a little bit different staining, and interterritorial matrix which fills the space between neighboring cells. And after this, this cell goes deeper in the cartilage, and it undergoes some mitotic divisions. Here we can see mitotic divisions of the chondrocytes, and then they form isogenic group of the cells inside the cartilage. Iso, it means same, genic, origin, so a group of cells with same origin which derive from one maternal cell. It's called isogenic or isogenous group. It's group of cells, cartilaginous cells, chondrocytes, which derive from one pre-existing chondroblast. They produce territorial matrix which surround themselves, and they maintain the homeostasis of the cartilage. So here we can see chondrocytes in the lacuna. And let's look at the cartilage. So this lacuna is present in deep parts, and we can see a superficial part of the cartilage. It's covered with chondroblasts. They cover the cartilage, and then they are going deeper. They become to be chondrocytes, which undergo some mitotic divisions, and they form isogenic group. And here we can see that in deep parts of the cartilage, isogenic group, they become to be bigger and bigger. It's growth of the cartilage, which is called interstitial growth. Here we can see interstitial growth of the cartilage when isogenic groups are growing. They enlarge their volume, and it's interstitial growth. And also another type of the growth of the cartilage, it's oppositional growth, when cells which cover the surface of the cartilage, they are growing, and it's called oppositional growth. It's growth provided by growth of chondroblasts, which produce new matrix, new interterritorial matrix on the surface of the cartilage. And cartilage is growing in the peripheral parts, this growth is provided by growth and production of matrix by chondroblasts. And also there is interstitial growth, which is provided by chondrocytes inside the isogenic groups. So two types of growth, oppositional and interstitial. It grows inside, growth of the inner parts of the cartilage. So two types of growth. And difference, you should remember difference between chondroblasts and chondrocytes. Chondroblasts, they are located on the surface of the cartilage. They produce interterritorial matrix, which fills the spaces between groups, and they aren't surrounded with matrix. They are located on the surface. They are free cells. And cells which are located in lacuna, they are called chondrocytes. They are mature cells. They maintain the homeostasis, but they have ability for the mitotic divisions. They undergo some mitotic divisions, and they form isogenic group. Here we can see isogenic groups, and each group includes the cells which derive from one pre-existing chondroblast. So this cell, for example, it goes deeper, and it forms a new isogenic group. It's growth and maturation of the cartilage. And also another feature of the cartilage is that cartilage hasn't blood vessels. Here we cannot see any blood vessels, and cartilage receives nutrients via diffusion from the surrounding tissues. For example, from the underlying bone or from the perichondrium, which covers the cartilage. So there are two ways how cartilage receives the nutrients: from the perichondrium, from the surface, and from the deeper parts, from the deeper tissues which underlie the cartilage. So here we can see growth of the cartilage. Here we can see chondroblasts. They are free cells. They are located on the surface of the cartilage. They aren't located in lacuna. Then they begin to produce matrix which surrounds them, and they surround themselves. They form own lacuna. And when cell has surrounded itself, it is called chondrocyte. So chondrocytes, they are located in lacuna. They haven't place where to produce matrix. That's why they don't produce matrix. Chondrocytes, chondroblasts, they have free space to produce matrix, and chondrocytes, they are already surrounded with matrix. And here we can see chondrocyte, and it undergoes some mitotic divisions, and it forms isogenic group or isogenous group, and all cells of this group, they derive from one pre-existing cell. And here we can see drawing of the histological slide, and it's cartilage. It's typical structure of the cartilage. So peripheral part, it's called perichondrium. It's dense connective tissue. It has two layers. Outer layer, it's fibrous layer. Here we can see fibrous layer, and it has a lot of collagen fibers. They provide protection of the surface of the cartilage. And inner layer, it's chondrogenic layer, or layer of stem cells. There are chondroblasts and also chondroprogenitor cells in this layer. They are located here we can see chondrogenic cells. They are located here in this layer, and they provide growth and regeneration of the cartilage. So here we can see mainly chondroblasts. They have spindle-like shape. And later they are going deeper. They surround themselves with matrix, and then they form isogenic groups which are growing. Number of cells is increased, and also cells, they increase their volume, and it's called interstitial growth of the cartilage. And this growth, which is provided by chondroblasts, it's oppositional growth of the cartilage. It's territorial matrix between the cells. It has violet shade, it has intense staining, it's darker, and between the isogenic groups, we have interterritorial matrix, which fills the spaces between neighboring isogenic groups. And here we can see perichondrium with chondroblasts and interterritorial matrix, and also there are isogenic groups developing. And here we can see also perichondrium on the microphotograph, different chondrogenic cells in the inner layer of the perichondrium, and deeper they become to be more round and they form lacuna which contain chondrocytes. Okay, so let's go to the main components of the cartilage. Here I can see main components of the cartilage matrix. You know that cartilage, it has cells, chondroblasts and chondrocytes, and also chondroprogenitor cells, and matrix. Matrix, it has fibers and ground substance. In the fibrous connective tissue, there are three types of the fibers: collagen, elastic, and reticular. In the cartilage, there are only collagen and elastic fibers. Reticular fibers are absent in the cartilage. So it's similar with connective tissue, but there is one exception: reticular fibers are absent in the cartilage. And different fibers in the cartilage, they prevail in different types of the cartilage. So there are collagen and elastic fibers, and they may prevail in different types of the cartilage. So here we can see cartilage. There are cells, chondrocytes, chondroblasts, and different directions of the fibers in the cartilage. And also spaces between the fibers, they are filled with ground substance. Ground substance of the cartilage, it has a lot of similarities with fibrous connective tissue. Ground substance has water, it has ions, proteins of blood plasma, its fluid part, and also there are GAG, glycosaminoglycans, which provide hydrophilic properties of ground substance, and they provide density of the ground substance of the cartilage. So ground substance of the cartilage is very similar with ground substance of the fibrous connective tissue, but it's much more denser. It has a lot of GAGs, more than in fibrous connective tissue, and they provide bigger hydrophilic properties of the ground substance, and that's why this substance is much denser. So there are GAGs, which include proteoglycans, glycoproteins, different complex substances which include proteins and also carbohydrates. And let's look at this structure of ground substance. So there are GAGs, they are main structures of the ground substance of the cartilage. There are unsulfated and sulfated GAGs. Here we can see unsulfated OH, here we can see unsulfated SO3. It's main difference between them. And unsulfated GAG is hyaluronic acid, and sulfated they are dermatan sulfate, keratan sulfate, heparan, chondroitin 4 and chondroitin 6 sulfate. So they are sulfated GAGs and unsulfated. And let's look at their relations. So here we can see hyaluronic acid, it's core molecule of these complexes, and there are linker proteins which are attached to the hyaluronic acid. And also there are core proteins which are attached to the linker proteins, and there are sulfated GAGs, they are different, they are attached to the core proteins. So like a branch of tree, and these complexes, they have similar structures. And here we can see hyaluronic acid, linker protein, core protein, and sulfated GAGs. And those sulfated GAGs, they have very big hydrophilic properties, and they attach molecules of water. Here we can see molecules of water which are attached to the sulfated GAGs. So here we can see structure of this complex: hyaluronic acid, linker protein, core protein, sulfated GAGs, and water. And this structure, it forms jelly-like substance, jelly-like colloid substance, which provides density of the cartilage. It's very hydrophilic, and it maintains a lot of water, and it provides the properties of the cartilage. And here we can see hyaluronan chain. There are linker proteins, core proteins, and also sulfated GEGs. And here also we can see hyaluronic acid, link or linker proteins, core proteins, sulfated GEGs, and they maintain a lot of water. They uptake the water, which provides hydrophilic properties of the cartilage. And let's look at the histological microphotographs. So here we can see isogenic groups. It contains chondrocytes and its territorial matrix between the chondrocytes. And also there is interterritorial matrix which fills the spaces between the isogenic groups. Here we can see peripheral part of the cartilage. It's fibrous perichondrium and chondrogenic perichondrium or layer of the stem cells. And then they give rise to isogenic groups. Here you can see deeper part of the cartilage. And in this place we have also bony tissue which underlies the cartilage. So here you can see structure of the peripheral zone of the cartilage. So there is peripheral layer which is called perichondrium with chondroblasts, and deeper we have lacuna with chondrocytes, and spaces between them are filled with matrix. So cartilage is a vascular connective tissue. It hasn't blood vessels. It receives nutrients from underlying tissues, for example, from underlying bone, and also they receive nutrients via perichondrium. Perichondrium also has blood vessels. It's only place where cartilage may have blood vessels. And nutrients they pass through the perichondrium via diffusion, and they are uptaken by the cells of the cartilage. And here we can see growth of the cartilage. It's perichondrium, peripheral part. There are chondrogenic cells, and they are giving rise to fibroblasts, and then they become to be fibrocytes. Chondroblasts, and they become to be chondrocytes, and they produce form isogenic groups which are growing, and it provides interstitial growth of the cartilage. Oppositional, it grows in the peripheral parts of the cartilage, and interstitial growth, it grows inside the cartilage. Here we can see places where oppositional and interstitial growth, types of growth of the cartilage are provided. It's a region of the oppositional growth of the cartilage, and it's interstitial growth of the cartilage. Cartilage grows from inside, and oppositional growth, it grows from outside in the peripheral parts of the cartilage. And now we are going to the classification of the cartilage, of the cartilaginous tissue. We have discussed typical structure of the cartilage, which is characteristic for all kinds of the cartilage. Now let's look what types of cartilage may be found in our body. We have three types of the cartilage: hyaline, elastic, and fibrocartilage. They have different properties. Here we can see hyaline, elastic, and fibrocartilage. Hyaline cartilage is the most typical type of the cartilage. It has perichondrium with collagen fibers. It has very thin collagen fibers which are almost invisible under the microscope, but they are present here. And elastic fibers, they are almost absent. Here we can see this typical structure, so it's hyaline cartilage. And also there are two untypical types of the cartilage. They are elastic and fibrocartilage. Elastic cartilage is very similar to hyaline cartilage, but there is one exception. Elastic cartilage has a lot of elastic fibers. Collagen fibers are almost absent, but elastic fibers prevail in this cartilage. They are well visible. They fill the spaces between chondrocytes, between isogenic groups. So in hyaline cartilage there are collagen fibers. They are very thin and they are almost invisible. And in elastic cartilage they are numerous elastic fibers, and they are well visible. And elastic fibers, they provide elastic properties of the elastic cartilage. Elastic cartilage is present in the nose, in the ear pinna, in the outer ear, and this elastic cartilage it provides elasticity of those parts of our body. And hyaline cartilage, it hasn't elastic properties, it's much denser than elastic. It's present in the bone junctions, in the articulations. It's present near ribs. Cartilaginous parts of the ribs, they are formed by hyaline cartilage. Also, hyaline cartilage is present in the trachea, in bronchi, and it's almost transparent. It's particularly transparent, and hyalus, it means glass, so it has glassy appearance. Hyaline cartilage, it's the most widely distributed type of the cartilage in our body. It's typical. And elastic cartilage isn't a very common type. It's present in those places of our body where elasticity and density at the same time are required. And third type of the cartilage is fibrocartilage. Here we can see this type. It hasn't perichondrium, first feature, and another feature, it has a lot of collagen fibers. Hyaline cartilage also has a lot of collagen fibers, but they are very, very thin and they are almost invisible here. But in fibrocartilage, collagen fibers, they are very thick, they are very numerous, they form bundles, and they are well visible in fibrocartilage. So fibrocartilage, it's a little bit similar to tendon, but it has much denser ground substance. And fibrocartilage is present in intervertebral disc, and it provides also strong properties of those discs. Hyaline cartilage, it provides density, and fibrocartilage also it provides strong mechanical properties of those parts. So it's stronger than hyaline cartilage. It's less dense, but it's stronger because of presence of big amount of collagen fibers. So there are three types of the cartilage: hyaline, elastic, and fibrocartilage. Hyaline and elastic, the main difference is elastic cartilage, it has a lot of elastic fibers and it has elastic properties. It's flexible and elastic. And fibrocartilage, it hasn't perichondrium and it has a lot of collagen fibers. It's stronger than hyaline cartilage. So there are three types, and let's look at them. So here we can see collagen. Here we can see hyaline cartilage, the most typical and the most common type of the cartilage. It has well-developed perichondrium. It has chondroblasts in the perichondrium. It has chondrocytes. But here we can see in the matrix we cannot distinguish any fibers because they are very thin. There are collagen fibers, but they are very thin and they have glassy appearance in this type of the cartilage. And here we can see hyaline cartilage. There are different chondrocytes in their lacuna, and there is a matrix between those cells. Also, here we can see hyaline cartilage. It's perichondrium. There are chondroblasts in the peripheral part of the cartilage and numerous isogenic groups which contain chondrocytes, mature cells of this cartilage. And here we can see also hyaline cartilage. So if you see this transparent amorphous matrix of the cartilage, you should decide that there is a hyaline cartilage. It's the most common and the most typical type of the cartilage. Also, here we can see hyaline cartilage. There are isogenic groups. It's perichondrium and spindle-shaped cells. They are chondroblasts, and chondrocytes, they are located in lacuna. Also, here we can see hyaline cartilage. It's perichondrium with visible fibers in fibrous perichondrium, and also chondrogenic layer with chondroblasts. And deeper part, there are chondrocytes in lacuna. They form isogenic groups. Places of localization, it's costal cartilage, cartilage of the ribs. Also trachea, bronchi, hyaline cartilage is present there. Also it's present in the articulations, in the bone junctions. And in the articular cartilage, it hasn't perichondrium, it's special feature. So perichondrium is absent in fibrocartilage, and also it's absent in the articular cartilage which belongs to hyaline. It's only type of the hyaline cartilage which hasn't perichondrium. And here we can see structure of the articular cartilage. It covers the bones, and it undergoes nutrition from two sources, from synovial fluid and from the underlying bone. It's articular cartilage, and it's covered with synovial membrane, which lines the cavity of the articulation, and it has three regions: peripheral region, intermediate, and deep region, which is divided into two: non-calcified and calcified cartilage. Calcified cartilage transits into the bone. So three regions: peripheral, intermediate, and deep layer. Deep layer includes a non-calcified and calcified zone. And also here we can see cartilage, superficial, transitional or intermediate zone, and deep zone, which includes non-calcified and calcified regions. And also there is tide mark or basophilic line, which separates two zones, non-calcified and calcified. It's structure of the articular cartilage, and you should remember features of this cartilage. It hasn't perichondrium, and it receives nutrients via synovial membrane, which covers it from the synovial fluid and from the underlying bone. But this type of the cartilage is hyaline cartilage, so it's special type of the hyaline cartilage which hasn't perichondrium. And one more place of this cartilage localization, it's epiphyseal disc. Here we can see epiphyseal disc, which is located near epiphysis and diaphysis. It's also made up of hyaline cartilage. Epiphyseal disc or metaepiphyseal plate, it provides growth of the bone in the longitudinal direction, and it's made up of hyaline cartilage. Here we can see epiphyseal disc. Here we can see this region. So you know from the anatomy that bones, long bones, they have epiphysis and diaphysis. And between those regions in children there is special layer of the hyaline cartilage which provides growth of the bone. There is layer of the resting cartilage or intact cartilage. Next zone is zone of proliferating chondrocytes. Here we can see chondrocytes which form columns. Here we can see them. They are undergoing mitotic divisions here, and it's region of the growth of the cartilage. Next, it's zone of chondrocyte hypertrophy. They enlarge their volume and they become to be round in shape. And the deepest zone, it's a zone of cell death. It's dying cartilage. Here cartilage is mineralized. And next, it's zone of the vascular invasion where blood vessels are growing inside the cartilage. So peripheral zones of the cartilage, they haven't blood vessels, and typical cartilage, it hasn't blood vessels. And deeper, there is a zone where blood vessels are growing inside the cartilage. So here we can see this structure. There are main zones where hyaline cartilage is present. So hyaline cartilage is present in the respiratory system, in larynx, trachea, bronchi. Also, it's present in the nasal septum, middle nasal septum. This cartilage is present in the ribs, cartilaginous part of the rib. Also, it's present in the articulation and in the in the disc, in the epiphyseal disc, which provides growth of the bone. And also there is cartilaginous stage of bony formation of bones in the embryonic period, and bones of the embryo, they are made up of hyaline cartilage. So almost everywhere hyaline cartilage is present. It's the most common type of the cartilage. And now let's go to the minor types of the cartilage. They are elastic and fibrocartilage, where they are present and what features do they have. So here we can see elastic cartilage. Here we can see numerous elastic fibers. It's special staining with orthene it was applied, and it reveals elastic fibers in this cartilage. Here we can see perichondrium with numerous chondroblasts which provide growth of this cartilage, and perichondrium has fibrous layer also. And here we can see isogenic groups of the elastic cartilage and very numerous elastic fibers. So orthene staining, it reveals elastic fibers, it reveals elastin, and we can see it in this type of cartilage. Without staining, elastic cartilage, it has yellowish shade, and hyaline cartilage, it has blue shade, and it's present in the nose. Outer cartilage of the nose, they are elastic. You can touch your nose and you can feel elastic cartilage there. Also, it's present in the outer ear, in the ear pinna. Also, it provides elasticity there. And some cartilages of the larynx, including epiglottis, they are made up of elastic cartilage. The main difference between elastic cartilage and hyaline cartilage is that elastic cartilage, it has elasticity and it has smaller density. It's not so dense as hyaline cartilage, but elastic cartilage, it's flexible. And elastic cartilage, it is never mineralized. Hyaline cartilage and fibrocartilage, they may be mineralized, they may undergo mineralization, but elastic cartilage is never mineralized. It hasn't opportunity for mineralization because it hasn't matrix for mineralization. Mainly collagen fibers are mineralized, they undergo mineralization, but elastic cartilage, it hasn't opportunity, it hasn't properties for the mineralization because it has mainly elastic fibers, and they haven't contact with collagen fibers. They haven't opportunity to be mineralized. And let's look at the histological slides. Here we can see elastic cartilage. Its outer layer, it's perichondrium and inner perichondrium. Its ear, ear pinna, and its section of the ear pinna. So outer and inner perichondrium, and between them there is deep layer of the elastic cartilage with cells of the elastic cartilage. There are isogenic groups of chondrocytes. And let's compare. It's a hyaline cartilage, also perichondrium on two sides and deeper parts with isogenic groups, and elastic cartilage. It has a lot of similarities, but the main difference is the presence of the elastic fibers. And we are going to the bigger magnification. Its outer layer, its perichondrium with fibers, its chondrogenic layer of the perichondrium with stem cells, and its deeper layer of the elastic cartilage with elastic cells of the elastic cartilage developing chondrocytes. Here we can see them, and these brown fibers, they are elastic fibers which are stained with orthene. And here we can see appearance of the elastic cartilage with different staining methods which were applied, and we have different appearance because different stains were applied. Here we can see hematoxylin and eosin staining, Welgert stain, aldehyde fuchsin, and elastic cartilage may have different appearance according to the applied staining method. Here we can see localization of elastic cartilage. It's external ear pinna, epiglottis, and also outer cartilage of the nose. So epiglottis and those cartilages of the nose, they contain elastic cartilage. It's never mineralized. That's why our ears and nose, they don't have bony density. They are elastic. They are soft if we compare them to bones and in other cartilages. And the basis of those cartilage is elastic fibers. And the last type of the cartilage is fibrocartilage. First feature, it hasn't perichondrium, and we can describe this type of cartilage as transitional from hyaline cartilage to the tendon. Here we can see particularly tendon, and it's hyaline cartilage, and it's fibrocartilage, region of the fibrocartilage. It has a lot of similarities with hyaline cartilage. Both types, they have collagen fibers, but hyaline cartilage has very thin collagen fibers. They are almost invisible in it. And in the fibrocartilage, we have bundles of the fibers, and they are very thick, they are well developed, and between the bundles we can see elongated chondrogenic groups. Here we can see them, and they have rows of the cells of chondrocytes, we can see them here, and bundles of the collagen fibers. Here we can see fibrocartilage. It's isogenic groups. They are elongated because they are located between bundles of the collagen fibers here. And also here we can see fibrocartilage. It's isogenic group and bundles of the collagen fibers. And places of localization of the fibrocartilage. It's pubic symphysis. Because fibrocartilage, it has strong properties because of presence of the collagen fibers. Also, fibrocartilage is present in the meniscus of some articulations, and also it's present in the intervertebral disc. Here we can see annulus fibrosus and nucleus pulposus, and this annulus, it's made up of fibrocartilage because it has a lot of collagen fibers and they provide this density of this structure. Here we can see fibrocartilage, and this annulus fibrosus, it's made up of fibrocartilage. It's main place of localization of this type of cartilage. And appearance of the fibrocartilage, bundles of collagen fibers and elongated isogenic groups between the bundles. And here we can see bone of the vertebra. It's a region of the hyaline cartilage, and deeper we can see fibrocartilage with collagen fibers. Here in hyaline cartilage we cannot distinguish fibers, they are thin, and here in the fibrocartilage we can see fibers. You can see fibrous structure of this region of the tissue. So here we can see fibrocartilage, it's hyaline and fibrocartilage. And deeper also we can see fibrocartilage of the intervertebral disc. Here we can see vertebra, thin layer of hyaline cartilage, and deeper we have layer of fibrocartilage. And here we can see hyaline cartilage and fibrocartilage, which is located deeper. It's fibrocartilage with bundles of the collagen fibers, and hyaline cartilage, we can compare them. It has transparent appearance, it hasn't visible fibers, and fibrocartilage, it has well visible fibers. And here we can see bundles of the collagen fibers and cells here. So it hasn't perichondrium, it has big density, but it has strong properties because it has big amount of collagen fibers, and fibrocartilage may be mineralized as well as hyaline cartilage, but elastic cartilage is never mineralized. So here we can see three types of the cartilage: hyaline, elastic, and fibrocartilage. They have a lot of the same features, but the main differences is type and amount of the fibers, and presence or absence of the perichondrium, and opportunity of possibility of mineralization. So hyaline and elastic cartilage, they have perichondrium. Fibrocartilage never has perichondrium. But there is one exception, articular cartilage, which is made up of hyaline cartilage. It hasn't perichondrium. Another difference: hyaline and fibrocartilage, they have collagen fibers, but in fibrocartilage, fibers are well developed, and elastic cartilage it has bigger amount of elastic fibers. And third difference is that hyaline cartilage and fibrocartilage, they have possibility of the mineralization, and elastic cartilage is never mineralized. Three main differences: presence or absence of perichondrium, type of the fibers, and possibility of mineralization.